published over 300 publications in high impact factor journals like Nature, Science, and Nature Chemistry. And he has received several prestigious awards. I will just name few, including Santi Swarup Bhatnagar Prize, one of the prestigious award in India in science field, and also JSPS. And if I will read his CV, it will take almost 1 hour because it's almost 36, 36 pages. So I'm just giving only few of them. And beyond his research contributions, Professor Mathi served as the editor-in-chief of journal Sinlet. And recently I got opportunity to interact and work with him. And he's very nice person, very humble and very, I can say, very helpful in nature and very helpful for the students. That is the quality. That's why he is successful in his research also. So please join me in welcoming Professor Mathi, sir. Over to you, sir, for your lecture. So please go ahead, sir. You can take 40 minutes, and 2-3 minutes we will take the question answer. So question hour. So 40 minutes, sir. So it's up to you, sir.
Thank you, Professor Dubeji. Thank you for that, very sweet and kind introduction. It's a pleasure to be here today. Thank you again for making me part of these great proceedings. I would have loved to be there in person, but I think this is still a great opportunity to interact with all of you. Let me try to share my slide. Hopefully it would be visible. Can you see my slides?
Yes, sir. It's visible.
All right. So great. Thank you very much again, all of you. Good afternoon. Hope you had a great lunch. It's a good time to fall asleep. If I can keep you awake, I would be very happy. I'm sure you had a great time. Well, I'm at IIT Bombay, as Dr. Abhishek Dubey was telling. I have had the great opportunity of having Professor Dubey over here with us. So thank you for visiting us, and thank you again for making me part of these great proceedings. Today I will discuss part of the ongoing research work in my laboratory. I think the best would be if you can keep your question coming. If you have any queries, feel free to ask. That way we can keep it more interactive. Okay. The area where we are working mainly is the carbon-hydrogen bond activation. Let's say if you take benzene, can you make aniline? If you take benzene, can you make phenol? Or if you can take toluene, can you make it hydroxytoluene? By doing so, you can definitely think of preparing a number of drug molecules, pharmaceuticals, agrochemicals, materials of interest. Other domain which we got excited about is the CH activation, let's say by photocatalysts. Can we react? Can we do the light-mediated reaction involving metal? Of course, while we are doing the photocatalysis, we thought that maybe we can do even the cross-coupling reaction. And since we want our reaction to be general and effective and greener and sustainable, we thought to also work on electrochemistry, flow chemistry, and biocatalysis. I'll come briefly to this point. And more recently, we have been also working on skeletal editing and CO2 valorization. Of late, we have been venturing it out in the domain of machine learning driven processes or so to speak, AI/ML, artificial intelligence, machine learning driven catalyst development. Okay. I'm sure many of us are synthetic chemists over here. We understand if we can start with simple molecules and we can convert into the complex one, that has many values. Also in our undergraduate studies, let's say bachelor or master's or PhD, whoever we are teaching, we realize depending on the electronic and steric properties of the substrate, we can do a desired functionalization. For example, you have known of Friedel-Crafts reaction, right? Friedel-Crafts alkylation, Friedel-Crafts acylation. It can be done at the ortho or para position. Now the question to you, to all of us, simply is, can we do a Friedel-Crafts reaction at the meta position of, let's say, toluene? Can we do a Friedel-Crafts reaction at meta position of toluene? The answer is not possible. So this is where, if we can attach ligand, these brown color entities are the ligand. So this is where we thought that we'll develop ligand which will be attached with the organic substrate and will be able to promote metallation at a position of our choice. For instance, if you have taken any of these brown color entity that you see over here and attach it with the organic molecule, it is possible to place the metal at the meta position. If we want to place the metal at the para position, we can do that with the help of any of these ligands. Now, of course, the first question that comes to mind, how these reactions are happening. Well, if you take an organic molecule, any organic molecule with a benzene ring, and if you would like to do a reaction selectively at the meta position, which in textbook of our bachelor or master studies may not be able to do because there is no selective catalysis available. Therefore, the reaction precisely at this position may be difficult. What we have developed simply is, you take your black color organic substrate, you attach this brown color ligand covalently. Due to the design of this brown color ligand, metal will attach with the ligand and the metal delivery will be precisely done at the meta position. This is similar to your pizza delivery, guys. You order a pizza or Swiggy or Zomato or Amazon. It is delivered at your doorsteps, not to your neighbors. Similarly over here, with the help of this brown colored ligand, the metal is delivered only at this meta position, not at the para position, not at the ortho position. Once the metal is delivered precisely, decisively at the meta position, and the metal is inserted at the meta position, you can do a series of reactions at the meta position. Of course, we have been developing this brown color ligand so that one can attach it with the organic molecule. For a variety of organic molecule, these metallation can be selectively done, as you see in here. The position is exclusively at the meta position. Now, you might will be asking, what if you want the metal at the para position? Because ortho position is relatively easy. For the para position, we have done the design. This whole designed ligand on the right hand side, that is over here. This is the ligand for para position. This biphenyl cyano over here, you see. Once again, with the organic molecule over here, you attach this right hand side, all this whole biphenyl unit. If you do that, once again, the pizza is delivered precisely at the para position. Metal is delivered precisely at the para position, not at ortho, not at meta. And once the metallation happens, then you can do the reaction following organometallic chemistry precisely at this position. Now, of course, these reactions are absolutely selective at the position that we want. If we want at the ortho position, we can do it. If we want at the meta position, as you have seen in the previous slide, we can do it. If you want at the para position, we can do it. One major drawback or the difficulty is you have to attach the ligand covalently with your organic molecule. And also, usually, of course, you need a catalyst. Let's say palladium is the catalyst here. You also need silver salt for the regeneration of palladium 0 to palladium 2. So silver salt, something like 3 equivalent will be required, and also very high temperatures were required. So these 3 limitations still exist. This is where we thought, instead of taking the organic substrate, attaching it covalently, which we were doing for several years, we thought, can we not have an attachable, detachable template, come and go, rather than covalently linked? Here it is covalently linked. Here it is attachable, detachable. Can we do like that? Well, we were able to exploit this strategy where you take your organic molecule, you bring your ligand, you attach them temporarily, you do the reaction, and then you remove it. This is what technically similar to what you see in enzyme. It happens in our body. Let's say you take any medicine or any food in our, let's say, liver, it gets metabolized, it gets reacted. What happens with the medicine or organic molecule? So the organic molecule resides in a substrate binding pocket. And this is the substrate over here, the big substrate. And there is, let's say, this enzyme called cytochrome P450, where you have this iron center with porphyrin ring. It can functionalize a desired and only one position of the substrate. There are many enzymes in our body. This is how they function. You take your organic molecule, you bring your catalyst. In front of the catalyst, your organic substrate sits down, and a particular position is getting functionalized, right? This is just a cartoon diagram or one of the enzyme I'm talking. Similarly, being a synthetic chemist, what we want, our organic molecules should be placed in front of the catalyst, and every reaction should happen selectively and efficiently. Now in enzyme, these reactions are happening for one substrate, one functionalization, efficiently, very efficiently, but one substrate, one functionalization. Synthetic chemist, everyone who are attending today, we would know that we want every substrate, not only one substrate, every type of substrate, every types of position, every type of reactions. Because if we are able to do that, then only we'll be able to prepare medicines, agrochemical, perfumes, materials of our interest. Some of these could be life-saving, some of these will be life-defining. Enzyme is great. It can do one reaction, one position, one functionalization. And this is where synthetic methodologies are important. As I was telling you previously, we were taking this black color organic substrate. We were attaching this brown color template, and we were delivering the metal at a position of our choice. Very recently, what we were able to show, if we take this template like this or the ligand like this without covalent attachment, here is covalent attachment, we just cut it open and we rearranged it, and the organic substrate is really oriented in a way which we wanted. Through this anionic hydrogen bonding, this is interacting, and the metal is delivered due to the design of the ligand. It is delivered at a position of our choice. And therefore, this metallation is happening at this position where you see over there. Okay, So what you have seen over here, previously, we were able to do a reaction which is pretty effective and happening precisely the way we wanted. Okay. Very recently, what also we were able to see that we can design the ligand further, incorporate this hydrogen bonding motif with the organic substrate. The organic substrates undergo hydrogen bonding with the substrates. It places the organic substrate in front of the catalyst, just like what was happening in the enzyme. Overall, by doing so, we are now able to see that these reactions are happening very efficiently and the way we want. Okay, so I would like to see if any of you have any questions. Do you have any questions? Anyone has any questions or comments so far? Anyone having any question? Yes, please go ahead.
Right now, they don't have any questions, sir. Please carry on.
Okay, sure. Thank you very much. Okay, so what we are able to show over here that if you take the organic molecule and if you are able to place the organic molecule in front of the catalyst, the way enzyme was doing, it is possible to deliver the metal by the design of the ligand and place the metal at a position of our choice. By doing so, we can pick up the carbon-hydrogen bond either at, let's say, at ortho position, meta position, or para position, as you see in here. We can do that and we can undergo the functionalization of our choice. As I was trying to tell you that it is important to see that these reactions are done at room temperature. What we thought that we'll have a ligand metal complex which will absorb light, and from photo-excited state, this reaction could be happening. Previously, we were hitting these reactions from 90 degree to 130 degree centigrade. And these reactions were therefore giving side product due to the high temperature, such as to 130 degree centigrade. In this particular case, what we were able to showcase that we can take a suitable ligand where ligand metal complex can absorb light, and these complexes then can undergo carbon-hydrogen bond activation from a photo-excited intermediate. Upon metallation, coordination with the organic substrate, insertion, beta hydride elimination can give rise to the palladium 0. And previously, this palladium 0 to palladium 2 formation was happening in presence of silver salt, superstoichiometric amount of silver salt. The problem with this approach of superstoichiometric amount of silver salt simply is the silver salt will produce superstoichiometric amount of waste. Therefore, waste management could be a big problem. The cost of things can also be a problematic. What we were able to demonstrate now that indeed with simple photocatalyst, by engaging oxygen and getting the photocatalyst excited with the suitable light wavelength, we can oxidize palladium 0 to palladium 2. So we believe with light, therefore, two steps are influenced. One is the palladium ligand complex is absorbing light. Instead of doing the reaction from the ground state, photo-excited state is reached by virtue of light. And then that from photo-excited state, CH activation is happening. And therefore, at room temperature, instead of 130 degrees centigrade, 90 degrees centigrade, we can do the reaction. This way, we can cut down the reaction temperature. Another important drawbacks or limitation that existed since time immemorial, that this oxidation of palladium 0 can only be done in presence of silver salt, because palladium 0 to palladium 2 oxidation required silver copper combination or super stoichiometric amount of salt. Now we are able to do that with a photocatalyst, right, like Yosin Y, Rose Bengal, simplest, cheapest photocatalyst, little amount of these, catalytic amount of these, in presence of light and oxygen as the oxidant, we can do these reactions. Of course, a lot of these reactions, one can do the tritium leveling, deuterium leveling of the medicines, which are again can be life-saving medicines. We can utilize this leveling for figuring it out how and what happens to the medicine when we are consuming in our body. So it can act as a radio tracer. By utilizing this method, we can go ahead and figure out what happens to the medicine which we are taking in our body. So these metabolism studies, all of these can be followed through by utilizing the techniques that we are able to develop over here. Of course, another way to achieve the greener and sustainable chemistry is to do this reaction instead of thermal reaction, if we can do this reaction under electrochemical condition. In this case, what we realize that photochemistry, the previous cases, although the change of reaction mechanism was not happening, but over here, we see there is a change of reaction mechanism and ligands are involved in these processes. Ligand redox chemistry is also getting involved. And here it is not necessarily palladium 2,0 or 2,0 catalytic cycle. Here could be also palladium 2,3,4 oxidation state. And these can be achieved under electrochemical conditions. All right. In the next stage, I would like to discuss some of the complex molecule synthesis. You see, all of these are pretty exciting natural products. The highlighted bonds in red, we thought, if we can prepare it by CH bond activation, it would be very, very exciting. Indeed, what we thought that we'll take a carboxylic acid and we will try to see if we can do the carbon-oxygen bond formation between these two oxygen. Okay, so what we see that this carbon-oxygen bond formation is a very difficult step, mainly because although the activation will happen at the methyl position, not at the methylene, this is going to be the preferred intermediate. But from there, the reductive elimination or the carbon-oxygen bond formation steps are going to be very, very difficult. This step is highly energy demanding. So this step is not going to happen pretty efficiently. And therefore, what we see, this preferred intermediate, if we can convert into a non-preferred intermediate, such as the CH activation at this methylene center, then we would have an opportunity to perhaps form a beta hydride elimination intermediate, which can undergo and give you the product formation. So this is where we were able to do more of a Cartan Hammett type of situation, where a preferred intermediate is not going for the product formation, but this preferred intermediate is undergoing or achieving further non-preferred or difficult intermediate to form, and that then undergo beta hydride elimination and can give the product. Okay. So this is, I think, mainly I wanted to tell you today. What you see over here is, a series of complex molecule synthesis, many natural product synthesis by utilizing these methods. Okay, I think, I will stop here today, and I would like to thank all my co-workers and all my, funding bodies and the people who are really behind this work, my group members and BRC, DST, ANR, FCS, DRDO, ISRO for this project. Once again, I hope I'm able to tell you that by utilizing the catalyst design, you can reach out to the different position of aromatic substrate, and also you can reach out different position of the aliphatic substrate. Okay, so once again, I will summarize by telling you that if you want to take the organic substrate like this, you can take the organic substrate, attach the ligand, you can reach out the meta position. And if you take any of these ligands, which are commercially available, you can reach to the para position. Similarly, for aliphatic substrate also, you can do this functionalization in a selective fashion. Okay, so I will pretty much stop in here. Again, I would like to take this opportunity to thank all the organizers of FCS 2026, in particular, Professor Abhishek Dubeji, for inviting me and having me here today. I would be happy to have your questions. Thank you very much.
Thank you, sir. Thank you for your very nice presentation and informative presentation. I'm audible, sir?
Yes, sir. Yes.
Okay. So this session is open for question answer. If you have any question, please go ahead.
Yeah, I have two questions. I'm audible?
Sure. Yes, sir.
I have two questions. In one CH activation, you show palladium metal.
Right.
So can we use the same group, nickel and platinum?
Yes.
Place of palladium or not?
Technically, yes. Technically, yes, definitely it is possible. But what happens, these ligands that are designed, sometimes these are not the best ligand for those, let's say, nickel or platinum even. You may have to design the ligand. The principle remains similar, but the ligand designing can vary a little. Okay, but yes, it's a very important question. We can indeed do, but the designing sometimes has to be little bit modified.
Yeah. The next question is that in the photocatalysis in the JAX paper, you use palladium 0. So is this palladium 0 will affect with palladium nano cluster?
Yeah.
If you use palladium nano cluster in place of palladium 0, is it possible to influence the reaction? And what is the light here? Either visible light or UV light?
Okay, that's a very good question. Again, first of all, the light can be household light, just the light bulb at home and house that we use. This is just a white light, white LED or white light, the standard light that we are having. And more importantly, the palladium that we are using, we start with actually palladium 2. So palladium acetate. Okay. Palladium 0 is forming in situ in the reaction mixture. So therefore, doing this reaction in presence of palladium nanoparticle may not be feasible. One of the problem of this sort of reaction is palladium nanoparticle formation, because palladium nanoparticle will not be doing the type of reaction that we are doing here. Therefore, we will have difficulties in doing with palladium nanoparticle. We are using mostly palladium acetate, palladium 2. This is palladium 2 starting material to start with, but as it goes through the reaction, palladium 0 is formed. Okay, and then this palladium 0 is oxidized to palladium 2.
Thank you. Thank you.
Sure.
Any more question? Anyone? I have, sir, one question.
Sure.
It's not question, sir, just for my knowledge. I want to increase my knowledge, especially related to cross dehydrogenated coupling. What is the key to design ligand, especially for distal cross dehydrogenated coupling, sir?
Okay. First of all, for cross dehydrogenated coupling, we need to have CH activation from both the substrates, right? If the substrates are biased, then it is easy. That means only one positions are available for both the substrate, they would go ahead and do it. But if substrates are unbiased, then you need to have a directing group which can direct at a position of your choice. Now, if you want both the substrate to be unbiased, that is going to be very difficult, because then you have to have two different directing group or ligand which can do the activation of two different substrates. Usually, so far, people are doing one unbiased and another biased substrate. And that is how the cross dehydrogenated couplings are done. But if you want to do both the unbiased thing, which would be very good, but it would be requiring designing of two ligand or two ligand metal complex system which are orthogonal with respect to each other in a way so that it can work on the two different substrate in a simultaneous manner and efficient manner.
It's quite challenging, sir, especially for last one.
Right.
Thank you, sir. Thank you for no more questions. So let me thank Professor Mathi for very informative lecture. And I would like to request Professor Tarun, sir, please come here and introduce our next speaker.
Thank you. Thank you, Dubey-ji. Thank you, Professor Dubey.
Thank you, sir.
Yes. If you want, you can use this one.
Hello, can you hear me? Okay.
Yeah, I can hear you.
Professor Goel, are you there?
Yeah. Can you hear me?
Yeah. So let me introduce our next speaker, Professor Atul Goel. He's the Chief Scientist and Head of the Industry Research Alliance Group at CSI, Central Drug Research Institute, Lucknow. He is also serving as the Area Coordinator for Musculoskeletal Health and Disorders at the same institute. So he has over 20 years of research experience, more than 125 peer-reviewed research articles, and also hold 18 international and national patents. A significant aspect of his career is successful licensing and transfer of 6 technology to industry. These include indigenous fluorescent probes for COVID-19 detection, the green R nucleic acid staining dye, and diagnostic kits for arbovirus like dengue and Zika. He has got many awards, including CRSI Bronze Medal, Vasvik Award, and also Professor Asima Chatterjee Award. He is a fellow of Indian Academy of Sciences since 2021. So let's welcome him. His title is Multicolored Indigenous Fluorescent Probe for In Vitro Diagnostic and Biomedical Applications. So let's give him a round of applause. Professor Goel, stage is yours.
Yeah. Thank you, Professor Gupta, for kind introduction. So I'm just sharing my slide.
I see BCUC.
Can you see my slides?
Yes.
So my slides are visible. Hello. Can you confirm my slides are visible?
Yes, visual, sir.
It's visible, right?
Yes, sir.
Okay. Good afternoon to all of you. This is Atul Goel. Let me give a huge round of applause to the organizers, especially Dr. Shechi Mishra, who is my former PhD student. She joined my group in 2013. Till 2018, she was here at CSR CDRI. And I'm really happy to see the topic, particularly the frontiers in chemical sciences, where you are covering entire like energy, environment, and health. So today I'll be talking about multicolored indigenous fluorescent probes for in vitro diagnostic and biomedical applications. So before I start my presentation, I would like to tell about the CDRI where I'm working. It would be very useful for all the students and faculties, the kind of facilities that we have at CSR CDRI in terms of drug discovery as well as diagnostic research. So as you know, CSR comes under the Council of Scientific and Industrial Research, CDRI, which is a premier biomedical drug research institute, not only in India, but also in South Asia. And the mission of our institute is to discover, develop, and deploy therapeutic strategies to alleviate disease burden, particularly the diseases of national relevance. And the important mandate of the institute is to develop new drug molecules, to systematically explore the Indian flora and fauna for therapeutic potential, to develop diagnostic for detection of microbial and emerging co-infections like during this COVID time, and also to develop human resource specialization in the diverse area of drug discovery and development. So these are the activity portfolio of CDRI, particularly in the domain of research and development. So we are involved in developing new drug candidates. We have developed indigenous process technologies for APIs and for other important key intermediates, and also involved in diagnostics. So we have a strong team of biologists. Basically, more than 80% of the scientists in CDRI are biologists, covering the entire preclinical related research work. And we have a strong team of medicinal and process chemistry at CDRI. I have already mentioned about doctoral and postdoctoral training. So anybody who's interested to join CDRI, they can go to the website and see the details. And we have a very strong technical services like drug testing lab, GLP facilities, biological screening, and sophisticated analytical instrument facility at CDRI. So CDRI, in that CDRI, which is an integral center for drug development. So mainly we are working on 8 different project areas called microbial infection, parasitic infection, viral infection, cancer, metabolic, endocrine, neurodegenerative, and reproductive. If you talk about, so mostly we are covering the entire portfolio of the disease area under one roof. So in this case, we are also in support with drug target and mechanistic studies. We have a different department called chemoinformatics, computational biology, and biochemistry and structural biology. And it is very important to have an integrated pharmaceutics and pharmacokinetics, which play an important role in drug discovery research. And we have animal studies at toxicology and experimental medicine department, and also some of the preclinical development with the partner clinicians. So everything under one roof, we have entire from designing a molecule till preclinical. We have all the departments and facility at CDRI. So these are some of the track records. We have discovered 30 new drug products, 6 diagnostics, and so on. All these technologies, more than 80 technologies, and several publications and postdoctoral training and all. So important part is this is called Saheli, which is a first non-steroidal once in a week contraceptive pill for women. We have developed a method for alpha-beta RT ether for Plasmodium falciparum malaria. We have a KeenMind memory enhancer, and this product has been marketed more than 7 countries. Recently, this Saheli, which is termed as a Centroman, called Chaya, the National Family Program, it has been introduced in recently in last 2-3 years. In recent findings, we have developed one indigenous RT-PCR diagnostic kit, particularly for COVID-19 during this COVID period. And we have marketed one DNA staining called GreenR, under the mission of self-reliant of Government of India. So as I just mentioned, this is a green R dye, which is developed in our lab only, which is marketed, and this dye is also available at GEM portal. And during this COVID time, we developed a universal quencher, which worked for the entire visible region. So till today, nobody in the world is having such kind of broad quencher which can work for entire visible region. So I'll be talking about a little bit on these quenchers and the development of diagnostic kits at CDRI. So this is a modular kit that we developed particularly for dengue, chikungunya, and Zika. This technology we have licensed recently to MyLab company, where all these components are developed at CDRI, and then it is totally indigenous kit that we are trying to develop. So they have, MyLab has taken the technology from us. They are preparing the CGMP batches, and soon will be commercialize this diagnostic kit in the market. So this was the technology transfer, as I just mentioned. So now I'll just talk about the research work and the projects that are going on in MyLab. So particularly, we are involved in developing some new leads from natural sources, particularly in the area of osteoporosis and bone-related disorders, where we have developed two interesting molecules called CDRI 399, which is a biodegradable bone implant material for bone regeneration. So I'll just devote to one slide particularly to explain about this. And another one is a CDRI 1500, which is a orally active fracture healing molecule and totally new chemical entities. So all these studies for this molecule was done from hit to phase 1 clinical trial, preclinical phase 1 trial. We did all this work in GLP at CDRI. And in another project, we are working on the development of diversity-oriented synthesis of new donor receptor organic fluorescent molecule, and for their application in developing new diagnostic and for biomedical research. We are developing some fluorescent dyes. So we have developed some DNA staining dyes, lipid droplet markers, vacuole staining dye, and neuronal cell body dyes. So these chemicals have been synthesized in our lab. We did all this biomedical application, and then some of the products are in the market. And in chemical sensors category, we have hydrogen sensors, and we have also developed some biometal sensors, particularly for zinc, iron, calcium for biomedical research work. So as I just mentioned, regarding this 399, which is a new medicated bone implant material to accelerate the fracture repair. So if you see that if there is a accidental fracture and you have a broken bone, so mainly the doctor fixes the bone with the metallic rods, pins, and screws. But the only problem is that they are non-degradable bone implants. So the disadvantage is a high rigidity, which causes the stretch shielding, and it inactivates bone metabolism at the fracture site. So when you put a kind of screw, stainless steel screws or pins, particularly on bone, so at that fracture site, there is no healing at all because it's a metallic implant. And it also interferes with the radiological imaging called CT and MRI. And sometimes it causes foreign body reactions and allergies, which are really, really difficult to treat using this IV treatments or antibiotics or so. And you know that after that, if there is a recovery, then patient has to go again to the hospital to take this metallic implant back. The surgery, the second surgery is also required particularly, and you can see the huge trauma to the patient. So what we come up with a new idea that if we can have a biodegradable bone implant material where we have a magnesium strontium based alloys in combination with our osteogenic molecule, which is 399 molecule. And in that combination, if we can prepare a kind of screws, pins, and rods, that would be a biodegradable, which has additional advantage of excellent biocompatibility, no interference with the CT MRI, and coated compounds with 399 will increase the bone formation at fracture site. And the important part is that these magnesium and strontium alloys were taken in a sense that they also show some osteoconductive property. So the healing will be more faster compared to in the normal cases. And if it is a kind of biodegradable, then you need not to go for the second surgery. So that was the technology we transferred to Orthorogenics Private Limited. And for this, we got CSR Technology Award by the former President of India. So I'll just show how these biodegradable screws and pins work. Just quickly, I'll just show you. So hope you have seen how it works actually, the biodegradable bone implant materials. So in another molecule, I just mentioned there's a new chemical entity, CDRI 1500. This is a orally active fracture healing molecule. So there is a huge medical need as there is no FDA approved oral drug in the market worldwide actually. People take only the calcium supplement and it auto heals. So we got one interesting lead molecule from natural product from Butia monosperma, which is called as a tar. And in that we got one interesting molecule called medicarpine, which shows a very interesting osteogenic as well as anti-osteoclastogenic property. So with that molecule, we could able to develop more than I think 200 to 50 compounds by putting some different functionalities, typical medicinal chemistry approach, and we could identify this 1500 molecule, which is orally active and it enhances the bone regeneration, particularly at 1 milligram per kg dose. So you can see the dose is very, very less in this case. And new bone formation at the fracture site was increased by 40 to 50%. So in general, suppose if auto healing takes a month or so, and if there is a 50% fast recovery using this 1500, so within 15 or 18 days, that patient can go back to the work, normal life. So this technology we transferred to Trica Pharmaceutical in Ahmedabad for clinical trials, particularly phase 1, phase 2, phase 3. So they are taking care of the entire phase 1, phase 2, phase 3 at their own expenses. And recently they got CDSCO approval for first in human trial. So in May 2025, they have already prepared CGMP batches, 3 different CGMP batches. Stability studies has been done, tablets have been formed. And now within 2-3 months, there will be a first in human trial at Vida Center in Ahmedabad for this compound. So I'll talk about this fluorescent probes in diagnostic, particularly in the area of RT-PCR based diagnostic kits for COVID-19. I think this slide doesn't need any introduction because in January 2020, the first case of COVID-19 infection was reported in Kerala. And it was declared the pandemic by World Health Organization in March 2020. And our Honorable Prime Minister imposes a national-wide lockdown on March 25th in 2020. And these are on the top right-hand side, you can see these different variants. The initial was alpha, beta, gamma, delta, and omicron. That was the last one. And this is the genome structure, particularly the target gene of RT-PCR based detection of SARS-CoV-2. If you see the structure and genome organization, the virion is covered with the spike protein as well as the membrane and envelope proteins. The genomic RNA is surrounded by N-phosphorylated nucleocapsid proteins. And the SARS-CoV genomes, which contains 29,903 nucleotides. The important primers and probe set target for the different regions of SARS-CoV-2 is a ORF1, AB, envelope gene, E gene, N gene, S for a spike, and RDRP sequence. So I don't know whether you have remembered that whenever you give these swab samples for COVID testing, so these genes were detected in your swab samples. So one of these E gene and gene were detected positive. If they are positive, that means the patient is COVID positive. So what we thought that during this mid 2020, when there was several cases of COVID-19, the RT-PCR cost per sample was 4,000 to 5,000, which was very, very high, and it was not affordable by the population of India. So what if we dig into it, we found that why this cost per sample is so high. So when we found that there was a supply chain disruption for these TechMan probes, which are important integral part of RT-PCR based diagnostics. So in the quench state, we need a quencher at 3' end, and at 5' end, we need a fluorophore. And this part belongs to a particularly for COVID-19 gene sequence like E gene, N gene, and RDRP kind of gene that I talked about. So during a normal state, it exists in a quench state. There is no fluorescence. When during this primer annihilation and probe hybridization, the sequence primers get elongated, and during this extension reaction, this fluorophore comes out. So that fluorophore, if it is attached with a green fluorophore, you will get a green emission at particular wavelength in 500 to 530. And then you can detect whether the patient is positive or not. So with this concentration, what we thought during the COVID time, nobody was allowed except those who are working on COVID related work. So and during this whole COVID period, we were in the lab working with all the students for developing this RT-PCR diagnostic kit. So this is a TechMan-like probes, and so far still people are using the imported TechMan probes. So we are the first one in India who can develop this dual labeled probes using our own quenchers and fluorophores. So these were the commercial dyes required particularly for, let's say, E gene, N gene, S gene, and RDRP. And similarly, you need a compatible quenchers. These are black hole quenchers. We developed our own quenchers. A single quencher can work for the entire visible region. So we filed this patent as well in India as well as abroad. And as I just mentioned that during this COVID time, we developed all these different colored dyes, particularly at CDRI only. These are some of we benchmarked it with the commercial dyes, like mentioned here. This is a FAM dye, fluorescein dye, which is commercial ones. The spectra is like here, and this is the dye that we developed. We put instead of phosphoramidite chemistry, we use azide so that the stability will be higher, and it will be very cheaper compared to the commercial dyes. Similarly, we developed another colored dye called Psi3, and we benchmarked it. Furthermore, we did in the red region, we developed this calfluor red, and then the last one was Psi5. So once we have this type of dyes in hand, these are the black hole quencher. Traditionally, 2 quenchers are required for doing multiplexing or triplexing. We could able to develop one interesting quencher called CDRI quencher. Is right here. You can see this is the commercial one. The absorption band is just double in this case by putting some functionalities on it to tune this photophysical properties of this quencher. So once we had this quencher in hand, we have to attach it with on solid support called CPG support. We did coupling reaction at this, and then using this DNA synthesizer, we could able to develop dual labeled probes where we developed this kind of fluorophores with azide. We used at alkyne terminal using our CPG quenchers, particularly for RDRP, E gene, and N gene, and did the click chemistry to develop a in-house dual labeled TechMan probes. We did HPLC purification. We identified this we purified this oligo, and then later on this pure oligos were tested in the real patient samples by my colleague Dr. Niti Kumar. These are the COVID positive patient samples, and you can see the nice spot curve. This is a FEM E gene, particularly attached with FAM, RDRP gene attached with Texas red. So different colors, you can see this one is for green, this one is for red. If these colors are showing in that region, you will say that the patient is positive. And we want to check the sensitivity of these primers and probes. So at higher CT value, that means low viral load, our primers probe work very well. You can see the nice chord curves in RT-PCR system. So it is important to have a third-party validation for this work. So we assembled this kit and sent it to CCamp Index for third-party validation. So independently, they studied phase 1 and phase 2, where they used 25 negative and positive samples. In phase 2, they have a 20 and 30 positive. And overall, they benchmarked with Q-line kit. This is a commercial die, and this is the die that CDRI developed in our lab. And we were happy to see that there was 100% concordance. The results were shared by them. And then later on, we assembled this kit for ICMR validation for approval. And we submitted this to NIV for validation. And then we were happy to see that there was 100% sensitivity and the specificity was 98.8%, which was benchmarked with this commercial ones as well. So we transferred this technology to Biotech Private Limited in Hyderabad. And this universal quencher was also, the technology was transferred to them so that they can manufacture it in India and commercialize in India as well as abroad. And this was highlighted during this COVID time that CDRI has developed this diagnostic kit. And within the 2-3 months, we could able to develop a specific kit, particularly for Omicron virus as well. And now we have a kind of platform technology. So we are ready for it. And in case of any emergent virus or comes, we are ready to tackle by using our own indigenous technologies, particularly for infections of emerging infections in future. In another project, we developed a DNA staining gel and a PCR master mix in collaboration with Biotech. So this was taken under the public-private partnership model. So I'm not going to go into this detail about the nucleic acid staining dye, which is nucleic acid plays an important role in transmission of genetic information. And these are some of these dyes like thiol orange, cyber safe, ethidium bromide, cyber green, and pico green. These are the commercial dyes available in the market. And this cyber green captures more than 80% market worldwide. So all these life sciences researchers who wants DNA staining dye, particularly for quantification and all, they are using cyber green, which is very, very costly. If you take 0.5 ml, it costs around 25 to 30,000. So what we thought that if we can develop the indigenous dye, and I have just already mentioned that 78% market is captured by cyber green, is right here. So what we thought that these are the two commercial dyes available, cyber green and cyber safe. We put some kind of functionalities to tune the properties so that it can bind efficiently with DNA. And we were lucky to have one product called GreenR, which was very cost effective. And you can see the compare, it is around 40,000 and 8,000 nearly. And what we could able to develop a dye which is cost effective around 5,000, and it is also available on Chain portal. So these are the salient features of GreenR. It is cost effective, is stable at room temperature, and this GreenR dye binds to nucleic acid of all kinds like genomic DNA, PCR product, plasmids, and RNA. And this is a totally indigenous product. Like as mentioned, that cyber green is an imported dye, which is costly, and we have developed this GreenR. And you can compare, this is the cyber safe dye, and this is the GreenR dye showing the bright gel images. We transferred this technology, and based on that, we could able to, like I already mentioned, this technology was transferred on National Technology Day. And we could able to launch this product in February 2023 in the market. So for the last 3 years, the sales is improving. They got repeat orders, so that means it is working very well. So finally, I would like to acknowledge what people see is a success. Like I'm telling about all these stories, but the important part, all these research fellows, you can see here, and Dr. Shachi Mishra is also in the team. and they have done all this work, particularly the hard work, persistence, late night, rejection, sacrifice, discipline, criticism, doubts, failures, and grace. So they are facing. So my whole acknowledgement. And finally, I would like to thank organizing committee for inviting me for this presentation, and I'll be happy to take any questions you may have. And finally, I would like, this is particularly for students. So don't be a parrot in life, be an eagle. A parrot's talk way too much, but can't fly high. But an eagle is silent and has the willpower to touch the sky. Thank you very much.
Thank you, Professor Goel. We have time for a couple of questions. Anyone from the audience? Or anyone online would like to ask question? Unmute. Okay, we have a question here. Go ahead.
Yeah, please. I cannot hear you, sir.
No, come here. Ask from.
Hello. Can you hear me?
Yeah, I can hear you.
So nice presentation, sir. I
Thank
have
you.
small questions.
Yeah,
What
please.
are the limitations of your kit which you have transferred the technology to one company?
Limitations in sense of what?
RT-PCR you had mentioned.
Yeah. So in that we have developed a totally the in-house Techno-like probes where we used our own quenchers and dyes developed at CDRI.
And how about your green R, sir? Is there any scope to improve further?
No, it is always, in science, actually, that is not an ultimate product. Since I have already mentioned that this dye is working very well in several institutes. So for the last 3 years, they have an increasing sale. So which means that people are adopting it, and they have done twice and thrice experiments, particularly at their research work, and they are very much satisfied. So we are trying to replace the commercial imported dye called cyber green with our own greener dye, which is totally indigenous in nature.
Okay, thank you very much, sir.
Yeah, thank you.
So thank you, Professor Goel, for a very informative and motivating talk. And on behalf of all this audience, I would like to give you a round of applause.
Thank you, Professor Gupta. Really happy.
Thank you so much. Thank you. So
Yeah,
that
thanks.
concludes our session. हम किसी को शेयर कर सकते हैं, माइक कर सकते हैं हमको
Is my voice audible?
Yes.
कोई response भी नहीं आ रहा उधर से. Hello? Hello? Is my voice audible? ये mic से बोलते हैं.
Yes, yes, voice is audible.
खोलिए. Class presentation.
पेन ड्राइव में है उनके, लेकिन उसमें सॉफ्टवेयर खुलेगा?
हाँ, ओपीएस में चलेगा, डायरेक्ट वहां से पेन ड्राइव लगाइए.
इसमें जूम बोलना पड़ेगा, फिर उसमें
किसका करना है लेक्चर?
उसमें जूम डाउनलोड किया हुआ है।
किसका लेक्चर करना है सर? अमित सर का.
हाँ,
तो पेन ड्राइव
पेन ड्राइव यहाँ तो हो ही जाएगा. यहाँ चेंज कैसे करेंगे?
साउंड तो किया रहेगा, साउंड कैसे चलेगा? साउंड कैसे चलेगा? साउंड कैसे चलेगा?
आप उससे कीजिए, उससे कीजिए.
करना पड़ेगा, बनाना पड़ेगा.
ओपीएस में
हाँ, दोनों पेन ड्राइव, बट सर पेन ड्राइव अरे बहन तो ये इसमें जाएगा नहीं. सी टाइप ओनली ना, थंडरबोल्ट पोर्ट चाहिए.
एक समस्या नहीं है,
क्या हो गया?
अरे उसमें जाता नहीं है, कन्वर्टर चाहिए.
मैं अपना लैपटॉप लेके आऊँ.
ओके ओके, ला रहे हैं, हो रहा हो रहा.
देख रहे हो ना क्या क्या व्यवस्था करना पड़ रहा है. ये डालो
आपके खुल जाएगा पीपीटी,
अलाउ करो, अलाउ
खुल जाएगा.
नहीं खुलेगा.
खुला ही कहाँ है?
यहाँ आइए. नो नॉट नाउ, नॉट सो बैकअप नहीं करना है. नॉट नाउ, नॉट नाउ, नॉट. आगे देखिए. हाँ,
कहाँ छोड़िए आप भैया, मेरे नाम से. ये नाम है,
अमित कुमार, ये रहा, मिल गया.
ये वाला है
हाँ मिल गया.
डॉक्टर अमित एट जेपी यूनिवर्सिटी. ये है डॉक्टर अमित जेट जेपी यूनिवर्सिटी.
कौन सा इसमें?
बुला लीजिए. हाँ नहीं जेपी यूनिवर्सिटी कौन सा है?
कितनी जगह दे चुका है भाई ये.
पूछिए, बुलाइए ना.
सर
सर एक मिनट, आप पीपीटी खोलिए.
कौन सा है सर इसमें? बस
And date wise.
हो गया हो गया हो गया relax.
Sort by
Date
date by.
date वो added date modified ऐसे कुछ लिखे दो.
Date modified full नहीं हो रहा है क्या?
एक सेकंड
Date.
ये ना
ये वाला शेयर करिए, पहले शेयर कीजिए। हो गया।
हो गया हो गया. इसको वहाँ शेयर कीजिए. अच्छा इसको यहाँ शेयर कर देना. यस क्यों आप मुझे होस्ट बना दिए. वो तो मैं पहले होस्ट बनाएगा तब.
ओके, बना रहा हूँ, चालू कर रहा हूँ।
ये जेपीयू FCS 2026 जेपीयू
Yeah, good.